Welcome to this video on prokaryote structure. I'm going to run through the different features of our prokaryotes here and tell you them exactly what they do. So a prokaryote is a simple organism that is different to a eukaryote, and it has special features that you'll need to know, especially for A-level biology, and you need to know exactly what they do. So let's start off right in the centre here with this big red squiggly line, which is the nucleoid or circular DNA. This is the genetic information that contains all of the genes which can be transcribed into proteins. The proteins can be transcribed on ribosomes and translated into polypeptides. So the ribosomes in a prokaryote are slightly smaller than the ribosomes you find in a eukaryotic cell, and they are size 70S. And this is where translation happens and protein synthesis there. These are usually free floating in the cytoplasm. Let's continue on with cytoplasm. So this is just the same as a eukaryotic cell cytoplasm. This is where a lot of metabolic reactions will occur, and likewise we're going to have inside that cytoplasm we have something called a plasmid. A plasmid is a circle of DNA, or a loop of DNA. This is a very useful entity because we can use this in genetic engineering, where you can cut out genes from that loop of DNA and then we can insert genes from other organisms so we can have them transcribed and translated. The reason they're used in genetic engineering is because bacterial cells divide very quickly, and it means we can have a duplicate of that gene within about 20 minutes each time. So that means we can now produce things on mass scale, such as insulin. The next thing we're going to look at is the layers surrounding our prokaryote. And let's start with the green layer here, which is the plasma membrane, also known as the cell membrane. This is a typical phospholipid bilayer structure. The next layer is the cell wall. The cell wall is quite similar to a plant cell wall, except it's made up from a different material, and this is called peptidoglycan, or often referred to as murin. Peptidoglycan is a type of carbohydrate. The next layer is the red layer here, and this is quite thick. This is known as the capsule, sometimes known as the slime capsule. And this is really there for protection and defense. The next structure we can look at is this tail here, which is also known as the flagellum or flagella. This is for movement. And the last structure here, we've got it listed, is the pili. The pili are structures which stick on the outside of your slime capsule, away from in the membrane here. You can see it's pointing outwards like this. And these pili help the bacterial cell to adhere to cells. If you get infected with a bacterium, it goes into the bloodstream. It can then adhere to a cell, stick to a cell using the pili, and it allows it to secrete toxins into that cell so it can damage it. The last thing that isn't listed on here is things called mesosomes, which are protrusions from the cell membrane. Mesosomes are the way that prokaryotes do respiration. There are no mitochondria, so mesosomes equals aerobic respiration. I hope that's been helpful on prokaryotic structure.